Welcome to module number 6, and today I will be discussing the biology of Old and New World primates. So let's get started. Let's start with a short but profound observation. We are primates. We share many of the same characteristics of other primates, like a large brain, grasping hands, generalized teeth, and strong mothering skills. It is important too to recognize that our close resemblances to many primates is not a coincidence. We are so similar to them because we are related to them. We did not come from them. We share a recent common ancestor with the other primates. More specifically, we share a very recent common ancestor with the African apes, a more distant ancestor with the monkeys, and an even more distant ancestor with the prosimians. Do not make the mistake of thinking that we study primates as living time machines to somehow go back in time and see how our ancestors lived. Primates today are as evolved as we are, but they do not provide us with examples of how a closely related animal has adapted to its environment, giving us insight into how our early human ancestors adapted to a quite similar environment. In this lecture, we will dive into the field of primatology. Hopefully, you will be able to quickly see why we study primates. We will examine what primates are, what features they have in common, and how one would identify a primate fossil. Finally, we will look at the different kinds of primates, from lemurs and baboons to chimpanzees and humans. Humans are primates. But what are primates? Primates are a group of closely related animals that live in the tropics. The map above shows the distribution of non-human primates found along the equator in South America, Africa, and Asia. Primates are arboreally adapted mammals, possessing many behaviors and anatomies that help them live a life in the trees. Primates also display remarkable dietary plasticity, eating a wide variety of foods, ranging from grasses and leaves to other animals. Finally, among the mammals, primates engage in the highest amount of parental investment, spending considerable time and energy in the few offspring that they have. Let's look in more detail at these characteristics and the adaptations that help primates survive. This figure has a lot of information on it, so we'll walk through it in detail. In the middle is a diorama showing the diversity of primates that can live in one forest in West Africa. The location of the forest is shown here in the map in the bottom right corner of the image. There are 13 different species of primates represented here, including humans. Notice that most of them live in the trees. Most live during the daytime, though others, like the small prosimians in the bottom right corner, come out at night. Primates have a generalized skeletal structure, giving them great flexibility when moving through the trees. Some species have evolved specializations, like the long arms of gibbons and the long legs of humans. Primates have dexterous hands, giving them grasping ability. This is enhanced by dermal ridges on their hands and feet, which make their sense of touch more sensitive. Primates also have forward-facing eyes, which gives them overlapping visual fields, a greater sense of depth, and thus enhanced 3D vision. But this comes at the expense of the sense of smell, which is reduced in most primates, and results in a shorter snout than what is found in other mammals. Primates have such a generalized tooth structure that they can eat a wide variety of foods. The teeth shown here are the upper and lower teeth of the red colobus monkey. Notice that the different shapes of the incisors, canines, premolars, and molars give primates the ability to choose many different kinds of food. Humans have the exact same number, 32, and kinds of teeth as other old world monkeys and apes. Primates tend to have very mobile joints, which allows them to navigate through the trees, at times in acrobatic ways. They have mobile shoulders and elbows, and quite dexterous hands and feet. An important primate character is the opposable thumb, which means that the thumb can touch the tips of the other fingers. This anatomy allows primates to grasp small objects like thin branches or insects. With our long, robust thumb, humans have the greatest opposability, and this has allowed humans to possess both a power grip like our ability to grasp and wield a hammer, and a precision grip, which allows us to pick up fine objects, like a pencil. A primate foot can sometimes resemble a human hand. In fact, some primates are called quadrumaneous, or four-handed, from the Latin quadri, four, and manus, hand. But primate feet are not hands, they are feet, like yours. Look at the chimpanzee foot skeleton and compare it to a human foot. Like us, chimpanzees and other primates have tarsals, metatarsals, and toe bones called phalanges. In fact, a human foot has the exact number of bones, 26, as a chimpanzee foot. These bones may be shaped a bit differently from yours, giving primates more flexibility in their feet, and grasping big toe or hallux, which allows them to climb trees easily. This is shown in the image at the bottom left of a young chimpanzee foot. Humans and many human ancestors lost their grasping toe as upright walking became the primary means of locomotion. Shown on the right is a primate vertebral column, which consists of 5 different kinds of vertebra: cervical, which is the neck; thoracic, which is the chest; lumbar, which is the lower back; sacral, which is connected to the pelvis; and coccyx, or the tail. Apes in particular tend to have vertically oriented spines, which is a pre-adaptation to human bipedality. Notice the S-shaped curvature of the human spine, which positions the head and hips in the same plane and balances the body in an upright posture. Notice too the long coccyx tail in the lemur spine. Humans and other apes have a short coccyx and lack an external tail. As previously mentioned, primates have an enhanced sense of touch compared to other mammals. There are sensitive dermal ridges on the tips of the fingers that give each of us a unique set of fingerprints. This sensitivity provides important information about the texture of objects, such as food or the branches many of our primate relatives walk on. In addition to these dermal ridges, primates are also equipped with fingernails instead of claws. Claws can be beneficial to some mammals, helping them cling like a sloth or grasp like a leopard, but they also can be a bit bulky and will get in the way of the fine precision grip primates use. Many primates require fine gripping of branches or precise grasping of the insects they eat, and have evolved a reduced claw known as a nail. Nails can be seen in this orangutan hand and of course are present on human hands as well. Compared to many other mammals, primates also have an increased reliance on vision. Primates have forward-facing eyes, as can be seen in this image of an orangutan. Forward-facing or convergent eyes allows the field of vision produced by each individual eye to overlap, giving primates depth perception and therefore 3D vision. This ability makes certain primates excellent insect hunters. Additionally, this depth perception is critical in an arboreal environment. Imagine leaping from one branch to another without being certain of the distance between the branches. In many primates, the eye orbit is also fully encased in bone, protecting this important sense organ. Color vision was an important adaptation in diurnal primates, who needed to be able to identify ripe fruit or insects during the day. The drawings on the left show an anthropoid primate, a gibbon, at the top, a prosimian, a lemur, in the middle, and a non-primate, raccoon, at the bottom. Notice that raccoons do not have any bones surrounding their eyes. Lemurs have a post-orbital bar, which partially encases the eye orbit. Anthropoid primates, like the gibbon or human, have an eye orbit entirely encased in bone. This is called a post-orbital closure. Of course, there are always trade-offs in biology. The enhanced sense of vision in primates have come at the expense of our ability to smell and hear. Notice the elongated snout of the lemur shown on the left compared to the reduced snout on the black and white colobus monkey shown on the right. Compared to other mammals, primates tend to have reduced olfactory or smelling abilities. Even within primates, the anthropoids, which are the monkeys and apes, have further reduced their reliance on the sense of smell. Look also at the nose and mouth of these two primates. Prosimian primates, like this lemur, retain a rhinarium, which is the wet nose that most mammals, like dogs, have. But anthropoid primates, like the monkey or like you, have a dry nose, separated from our mouths by an upper lip. So how do we explain the elongated snout on a baboon like the one shown on the far right? Do they have, in a sense of smell? The answer is no. That long snout has evolved to house the roots of enormous canines, or fang teeth, that help the adult males compete with one another for access to mates. Many mammal species have evolved quite specialized teeth. Primates, however, have retained the primitive mouth, with 4 different kinds of teeth: incisors, canines, premolars, and molars. This assortment allows primates to eat a wide range of foods. Not only do primates have these different kinds of teeth, they appear in very consistent patterns. Old World anthropoids, monkeys and apes, have a 2-1-2-3 dental formula. That means in each quadrant of their mouth they have 2 incisors, 1 canine, 2 premolars, and 3 molars. Check your own mouth. This is your dental formula, unless you have an impacted wisdom tooth or have had it extracted already. Now, New World monkeys and many prosimians have an extra premolar for a 2-1-3-3 dental formula. Tarsiers are like this too, but they have lost a bottom incisor for dental formula of 1-1-3-3 in the lower dentation. The image on the right shows a teeth from the side, or the left, and front, or the right, of 5 primates from top to bottom: human, chimpanzee, gorilla, orangutan, and baboon. Notice that although humans have different shaped canines, they have exactly the same number of each tooth type, and therefore the very same 2-1-2-3 dental formula as all of the other primates shown here. We will spend some more time on teeth. The reason for this detailed look at teeth is twofold. First, the numbers and shapes of teeth are very useful tools for distinguishing different groups of animals, and this includes the different primates. Second, teeth are some of the hardest objects in the body, and they therefore are preserved very well in the fossil record. Much of what we know about fossil primates is from their preserved teeth. An example of how teeth differ among the various primates is shown at the top. As we just saw, Old World monkeys and apes have the exact same number of teeth, where they have the same dental formula of 2-1-2-3. That means monkeys and apes both have 3 cheek teeth or molars in each quadrant of their mouth. So, if you find a fossil molar, how can you tell if it is from a large monkey or a small ape? One way is to look at the shape of the tooth itself. Monkeys have 4 high ridges on their teeth, 2 in the front and 2 in the back, producing what is called a biliophodont pattern. Apes do not. Apes have 5 low cusps that form a pattern that looks like the letter Y. This is called a Y5 pattern and helps us identify ape teeth. Notice the Y5 pattern drawn out from the left to right, the gibbon, chimpanzee, and orangutan teeth. If you look in a mirror, you'll see the Y5 pattern on your lower molars as well. Additionally, while most monkeys and apes have flat, vertically oriented incisors, most prosimians have combined their lower incisors into a scraper-like structure called a tooth comb, shown here at the bottom. A tooth comb helps these little primates extract sap from tree bark. One final aspect of primate dentition we'll examine is the canine. We've already seen that the canine tooth is much larger in the male than in the female in primates that engage in intense male-male competition for access to mating opportunities. Small canines, or canines that are not sexually dimorphic, tend to be found in primates that form long-term pair bonds, like gibbons or marmosets. These large upper canines, shown for instance on the yawning male baboon to the bottom right of this slide, serve to warn competitors and even predators. How do these primates keep the canine sharp as they get older, given that teeth naturally wear down over time? Primates accomplish this through something called the canine premolar honing complex. When a primate with a large upper canine closes its mouth, the upper canine slides into a space between the lower canine and the premolar called a diastema. This is shown on the monkey jaw in the upper left of the slide. Notice that the human jaw on the right does not have a diastema, or a large upper canine for that matter. When the canine slides into the diastema, the back of the canine rubs against the front of the sharp or sectorial premolar, keeping that upper canine nice and sharp. Humans, in contrast, rear the very tip of the canine when they close their mouths, something called apical wearing. This dulls the tooth over time, which would not be beneficial if we used our canines to ward off competitors. Let's now move on from primate teeth to primate parenting. Primate mothers are very dedicated. They give birth to few offspring and spread out their births so that they can invest time and energy into each of their babies. For instance, orangutans will have a baby and then nurse and care for it for up to 7 years before having another child. Chimpanzee mothers, like the one shown here, will go an average of 4 years between offspring. The time that mothers devote to their developing offspring is beneficial because of the important role that social learning has in improving a primate's chance of survival. Shown in the graph below is the growth and development of 6 different primates: lemurs, macaques, gibbons, orangutans, chimpanzees, and humans. Notice that apes tend to have an elongated life history. They have longer periods of infancy, longer childhoods, and even overall lives than monkeys or lemurs. Humans are the most extreme, having a very long childhood and overall lifespan. Related to this stretched out juvenile period is the opportunity to learn, and this requires a high level of intelligence. Elevated primate intelligence can be measured by the relative size of the organ of intelligence, the brain. Compared to other mammals, primates have large brains. The graph on the right compares sizes of the brain on the y-axis and mammals of different body masses, plotted on the x-axis. The average mammal is represented by the straight line. Notice that primates in general plot only above the line, meaning that they have larger brains than expected for a mammal of their body mass. Humans in particular have much larger brains than expected, and we can trace this increase in brain size, called encephalization, through the human fossil record. On the left are cartoon drawings of 3 different primate brains: a monkey and an ape, a chimpanzee. Notice that the brain of the chimpanzee is more folded and complex than the brain of the prosimian. Also notice the olfactory bulb, which produces smell, is reduced in monkeys and apes compared to prosimians. We have already discussed how the reliance on the sense of smell is reduced in anthropoid primates, and even the brain reflects this change. Notice, however, that the occipital lobe is increased in monkeys and apes. This is where visual signals are processed, and the enlarged size reflects the greater reliance on sight in monkeys and apes. The human primate is just one of over 200 different kinds or species of primates on Earth today, but we are not equally related to all of them. As we discussed in Module 2, primates, like other animals, can be clustered into closely related nested hierarchies. These hierarchies are shown in the diagram here, the primate family tree. By understanding the phylogeny, the family tree structure of living primates today, researchers can determine which anatomies unite particular groups. This knowledge is important because if researchers find a fossil with a particular anatomy, they can determine which primate group it is most related to. And they can also infer when and even why certain features evolved. We'll get into some of these features, like upright walking and non-honing canine teeth, in just a moment. For now, let's look at the family tree. The order primates can be split into 2 suborders, prosimians and anthropoids. Prosimians are often called lesser primates because they retain more primitive features than the more derived anthropoids. But do not make the mistake of thinking that living prosimians represent what the earliest primates looked like. Prosimians have evolved too, and some features of their teeth are highly specialized and derived. Interestingly, there are some features, like the absence of a wet nose, or the rhinarium, that unite one group of prosimians, the tarsiers, with the anthropoids. DNA evidence supports this grouping as well. Therefore, most researchers now subdivide primates into a group that includes the anthropoids and tarsiers. called haplorhines, by grouping all prosimians like lemurs and lorises, but not the tarsiers, as stepsirrhines. Anthropoids consist of the monkeys and the apes, but not all monkeys are equally related. The New World monkeys in South America, the platyrrhines, are less closely related than the Old World monkeys, cercopithecines, and apes, hominoids. These latter two, Old World monkeys and apes, are called catarrhines. Within the hominoids are the hylobatids, the gibbons, pongids, orangutans, and hominids, African apes, in humans. Let's look again at the different suborders of primates. In the last slide, I mentioned that researchers used to split the primates into two suborders, prosimians and anthropoids. This classification scheme is based on the fact that tarsiers are anatomically much more like lemurs and lorises than like monkeys and apes. This grade-based approach emphasizes the primitive anatomies that tarsiers share with other prosimian primates, such as a relatively small brain. However, instead of focusing on levels of anatomical complexity, more recent approaches have emphasized ancestor-descendant relationships and have found that tarsiers are more closely related to anthropoids than to prosimians. Under this evolutionary or cladistic scheme, tarsiers are united with anthropoids and collectively called haplorhines. The lemurs and lorises, which are the prosimians minus the tarsiers, are called stepsirrhines. The DNA evidence supports this classification scheme, as does the presence of shared derived anatomies found in tarsiers and anthropoids, including the presence of a dry upper lip or absence of a rhinarium. And these different classification schemes also have an important impact on how the hominoids are grouped together and named. Based on anatomical similarities and differences, researchers have historically grouped humans separately from the other apes. This is diagrammed to the left as the traditional classification. Gibbons had their own grouping, the hylobatids, or lesser apes. All of the great apes were thought to be closely related to one another in the pongid family, and humans had their own family, hominid. However, this has all changed with the use of DNA as a tool for figuring out whether this traditional classification was in fact correct. It turns out that the great apes are not all equally related to one another. In fact, chimpanzees and bonobos are more closely related to humans than they are to other apes. This new cladistic classification system reflects genetic similarity and groups humans and members of the genus Pan, which are the chimpanzees and bonobos, together in the subfamily Hominine. All of the great apes, including humans, are in the hominid family, and together with gibbons, comprise the hominoid superfamily. So what do we call just the humans and extinct human ancestors? Using the traditional classification system, they are called hominids. But in the cladistic classification scheme, the word hominid includes all of the great apes. So the word we use for just the human ancestors now is hominin. Before we dive too deeply into the apes, let's get back to those other primates, starting with the stepsirrhines. The primates least closely related to humans are the stepsirrhines. These are the lemurs, lorises, and gallicos. Stepsirrhine primates tend to have smaller brains for their body size than monkeys and apes have. They rely heavily on their sense of smell for finding food, and therefore tend to have longer snouts than monkeys and apes, as we have seen previously based on this image of a lemur. The fossil record indicates that the very first primates are very similar in many ways to modern stepsirrhines, as we will see when we start to discuss the primate fossils. Stepsirrhines are found today in Asia, Africa, and on the island of Madagascar. In fact, Madagascar was once home to one of the largest primates, a gorilla-sized lemur called Megalathivus. The image on the bottom of the slide shows how big these lemurs used to be. Identified in the image on the right are skulls of a mouse lemur and an indri, the smallest and largest lemurs respectively, on Madagascar today. The other skulls are from extinct lemurs, many of which died out when humans arrived in Madagascar over 1,000 years ago. Stepsirines and haplorhines diverged from each other at least 40 million years ago. This divergence was in part because each primate lineage evolved slightly different adaptations that allowed them to survive. Many of these adaptations persist today in the living haplorhines. For instance, most haplorhines, or anthropoids especially, tend to have larger brains and are more sexually dimorphic than the stepsirines. This means that the males are larger and have larger canines than the females. Haplorhines also have an eye orbit that is fully encased, or almost fully encased, as in the case of tarsiers, in bone, and can mostly see in color. But their enhanced vision has evolved at the expense of smell, which is more dominant in the stepsirines. Haplorhines also have a generalized diet and usually have fewer teeth than most strepsirines. Let's look at the haplorhines in more detail, starting with the most primitive and unusual of them, the tarsier. Tarsiers are very small primates found in Asia. They have very large eyes to gather what little light there is at night. They are nocturnal, and they are proficient hunters, eating a diet almost exclusively of insects. Their feet are also quite interesting and unusual. Tarsiers have very long bones in their feet, which provide excellent leverage for leaping through trees. Although these little primates are strepsirine-like in some ways, they also share derived anatomies like a dry upper lip, large canines, and large upper incisors, with monkeys and apes supporting their classification with the anthropoids under the umbrella term haplorhine. Remember, all primates can be divided into one of two groups, stepsirines and haplorhines. We've already discussed the strepsirines. All haplorhines can then be divided into two groups, a small group consisting only of tarsiers, which we just discussed, and a much larger group called anthropoids, monkeys and apes. All anthropoids can be divided into two groups, platyrrhines and catarrhines. We'll start with the platyrrhines. These are the New World monkeys that live in South and Central America. They are named platyrrhines because their noses tend to be quite broad. Platyrrhine means broad-nosed, with round nostrils and a wide septum between them. In contrast, catarrhines, which means hooked nose, tend to have nostrils that are close together and downwardly pointing as illustrated to the right. Platyrrhines can be identified in two distinct ways. First, they are the only primates besides humans in South and Central America. Second, they have a distinctive dental formula, 2133. Catarrhines, as we'll see, have one less premolar in each quadrant, a dental formula of 2123. There is a group of platyrrhines called ceboids, which include the cebid monkeys and the Adeline monkeys. One type of Adeline, called the woolly spider monkey, is shown in the upper left of the slide. As you can see in this image, these monkeys are more suspensory than most other monkeys, using their arms to support their bodies in a similar manner to how many apes move through the trees. They also make use of a fifth appendage, their tails. These monkeys have grasping or prehensile tails. Though many people think that all monkeys can hang by their tails, it is actually quite rare and possible in just a few species of platyrrhine. Catarrhines are monkeys and apes that live in the Old World, Africa and Asia. They first appear in the fossil record around 30 million years ago. Catarrhines diverged into two groups, monkeys and apes, about 25 million years ago. Old World monkeys, or cercopithecoids, are quite numerous and successful. They live in a wide range of habitats, have a diverse diet, and live both on the ground and in trees. Most cercopithecoids are quite sexually dimorphic. There are two different families of cercopithecoid, colobines and cercopithecines. Images A, B, C, and D across the top of the slide are colobine monkeys. Black and white colobus, gray langur, proboscis monkey, and dauc langur. The others are cercopithecines, mandrill, the Brazos monkey, olive baboon, and vervet monkeys. Colobines tend to live in the trees and eat mostly leaves. They also have sharp molars to slice into the leaves and often have a multi-chambered stomach to help with the digestion of the plant material. Cercopithecines have a more diverse diet consisting of fruit and thus have more rounded molars and lack the multi-chambered stomach. Many Cercopithecine monkeys have been studied in depth. These include baboons, geladas, macaques, and vervet monkeys. Many of these Cercopithecines live in savanna environments in East Africa and provide an interesting model for understanding the evolution of our own lineage, which once occupied a similar habitat. By understanding how these monkeys adapt to their grassland environment, researchers can gain some insight into how our own ancestors made the same sort of adaptation millions of years ago. Though Cercopithecoids can serve as a nice model for early human behavior, we are not Cercopithecoids. We are hominoids. Hominoids, or apes, are large-bodied, large-brained, tailless primates with broad faces and a Y5 pattern on the lower molars. Shown here are the different species of non-human hominoid. Across the top are from left to right chimpanzees, bonobos, and orangutans, and across the bottom, gorillas and gibbons. Keep in mind that humans belong in this grouping too. Chimpanzees, bonobos, and gorillas live in Africa, while orangutans and the lesser apes, the gibbons and siamangs, live in the forest of Southeast Asia. How can we identify an ape? There are a few ways. First, as we've already discussed, and is again illustrated across the top of the slide, apes have a Y5 pattern to their lower molars, quite distinct from the bilophodont pattern seen on Old World monkey teeth. In addition, the large male apes have huge chewing muscles that are attached to the top of the skull by means of a large ridge of bone called a sagittal crest. This feature appears in some human ancestors too. If you put your hands on the sides of your jaw and clench your teeth, you will feel your jaw bulge. This is the masseter muscle, important for chewing. Likewise, if you put your hands on the sides of your skull and bite down, you'll feel the temporalis muscle contract and bulge. Imagine if the muscle was not only on the side of your head, but was large enough to continue to the very top of your skull. The muscle would anchor to a ridge of bone across the top of the skull called the sagittal crest. That is a situation in many large modern apes, and even in some human ancestors as well. Therefore, by finding a skull with a sagittal crest, researchers can infer that these creatures had large chewing muscles and may have been eating leaves and plant stems, such as modern gorilla eat. Though there are clearly differences in the skull between apes and other primates, many of the differences actually reside in the postcranial skeleton, the bones below the neck. These bones are important for locomotion. The non-human apes all have very long arms and long curved fingers to help them navigate through the trees, as shown in the drawing of the gibbon skeleton on the right. These anatomies allow apes to engage in suspensory or arm-hanging postures, in which the body is positioned in an upright or orthograde manner. Gibbons are very suspensory, and these animals can fling themselves from limb to tree limb in a form of locomotion known as brachiation. The larger apes like chimpanzees, orangutans, and gorillas are too large to brachiate, and more often use their long arms and curved fingers to more cautiously move through the trees. The African apes, such as the chimpanzees, bonobos, and gorillas, will often come to the ground and move from food patch to food patch terrestrially. They tuck their long arms and fingers under their bodies in a quadrupedal form of locomotion called knuckle walking. This is shown in the image on the left, a gorilla bearing much of its weight on its knuckles. What is important to recognize here is that the human body plan generally resembles that of the African great apes. But while there are similarities, there are also important differences, and these differences allow us to walk on two legs more often and more efficiently than any of the great apes. For instance, as can be seen in the drawing on the left, the position of the hole in the base of the skull for the spinal cord, the foramen magnum, is toward the back of the skull in quadrupedal monkeys and apes, but at the bottom of the skull in humans. Keep in mind why this is important. If we find a skull, we can tell how it may have moved around its world by examining whether the foramen magnum is under the skull, as in upright walking humans, or towards the back, as in quadrupedal apes. The hip joint is also quite different in humans and in apes, and this results in small differences in the shape of the pelvis and the femur. Muscles that help extend the leg in apes are repositioned in humans, so they help balance the body when we stand on one leg. In fact, the next time you are walking around, put your hands just below your hip bones and you will feel these muscles contracting to keep the body from falling over when you stand on one leg during walking. And they contract on the same side of the leg as you're standing on. These muscles are crucial for balancing our bodies during two-legged walking, and they are able to do so because the pelvis and the femur are shaped a bit differently in humans compared to apes. If we find a fossil femur and it is shaped more like a human's femur, we can infer that these muscles serve the same role, and that the fossil is from something that walked on two legs, as we do. There is one last observation to make about these different ape skeletons. From left to right is a suspensory gibbon, an uncle walking gorilla, and an upright walking human. Notice that humans have differently proportioned arms and legs. The apes have very long arms compared to the length of the legs. Humans, in contrast, have much longer legs and shorter arms. Therefore, just the ratio of arm length to leg length in fossil bones can help us reconstruct whether an ancient primate was on the ground walking on two legs or swinging through the trees. We'll discuss these differences and how we can detect the evolution of these features using fossils in later lectures.